So thank you again. As I mentioned a few moments ago, my wife and I, the family, has a vacation place in New Mexico. And we love the Southwest, western part of the United States. And we became familiar with it as students when we would go on desert hiking trips. And we still do that. And on one hike not so long ago, we noticed this small plant. I recognized the plant. Its common name is the Colorado 4 o'clock, mirabilis. And I took a picture of the plant on the way out on our hike and then again on the way back. So the picture at the top was taken at 2 p.m. The picture at the bottom is taken at 6 p.m. And what I want to show you, notice this little branch here, so I'm not making anything up. It's present in both of the photographs. And you see on our trip out at 2 o'clock, the petals of the flowers are all closed. The blooms look nothing like functional flowers, but just 4 hours later at 6 o'clock, coming back from the hike, of course, this plant has come alive. Its blooms are open. So this is a rapid response in an arid environment. In thinking about how this response would arise in a plant, we understand that a moth, hawk moth, is the pollinator, the primary pollinator of this plant, and that's only active in the evenings and at night. And we're in a desert environment. Water's precious. It takes a great deal of water extended out into the petals to open those blooms. And so I suspect what this is an adaptation to conserve water in an arid environment, to open those blooms only at times when the pollinator is present and pollination can be successful. So this, for me, is paints an image of the fact that these rhythms, these biological rhythms, are all around us. You just have to open your eyes and take a look around your feet, and you'll see these even in the plants that you pass on a daily hike. Now, Carl von Frisch, who many of you may know, a Nobel laureate from the 1960s, spent his life studying the behavior of honeybees. And he made a very important discovery about their use of biological clocks. And we now know that the clock he was studying was a circadian clock, which is used in the dance language of the bees. Now, if you're unfamiliar with this system of communication in the bees, foraging bees will find food in the environment, come back to the hive, and the hive is dark. The comb is vertically placed, almost a dance floor, a vertical dance floor inside the hive. And the foraging bee will perform two kinds of dances, one of two kinds of dances if they've encountered food and want to communicate that to the rest of the hive. One of these dances is called the round dance, which is illustrated here. The forager, the communicating forager is just going around in circles. That simply means there's food close by. Just go outside the hive and find it. You can't miss it. A more interesting and complicated dance is this figure 8 dance called the waggle dance by von Frisch, that is performed by the forager. And that carries two components of information. One is the time that it spends going across the middle of this figure 8. It spends vibrating its wings and body, which is why it's called the waggle dance. And that conveys information about the distance to the food source. The other key component, though, is the orientation of this figure 8. So, for example, if the food source is in direct line with the sun, this dance will be performed such that the intersection of the two parts of this figure 8 is vertical within the hive. On the other hand, if the sun is off by a number of degrees, say 40 degrees off from the food source, the orientation of the dance will be shifted by the same number of degrees, so that anyone following this forager in the hive knows what direction, in which direction to move on leaving the hive. Now, von Frisch realized that this creates a problem because the sun is moving, or at least the Earth is turning to give an apparent motion to the sun. And the dances he was observing inside these hives were going on, in many cases, for several hours. So how do you communicate without going back outside and looking at the position of the sun? How do you stay inside the hive and nevertheless communicate the correct position of the sun? And what von Frisch realized was that the dancing bee slowly changes the orientation of the dance to correctly predict the new position of the sun. So we now know that this ability to calculate, to predict the position of the sun is due to a circadian clock. And Steve Reppert at University of Massachusetts Medical School has recently looked at migrating butterflies who also display the same kind of ability, and finds that in the antennae of the butterflies are cells containing clocks, circadian clocks, and that if you block the ability of those clocks to run in the antenna, the animals, the monarch butterflies in this case, will lose their ability to orient. So in this much more complicated piece of behavior, we once again see the animal calling on this ancient clock in a particular group of cells, not in the brain, but in the antennae. Now, perhaps much more familiar are our sleep-wake cycles, and these are, of course, familiar to ourselves, but these have been well studied, particularly in rodent models, in which animals housed in a cage with a running wheel for days and days in constant darkness gave the surprising result back in the 1960s, which is when this record is derived. The darkened areas, each one of these lines represents 24 hours, so we're looking at a record that's 25 days long. And you can see times when the animal's on the running wheel in these darkened areas. The darkened areas are produced by a pin that's driven by the motion of the running wheel. And so day after day, the animal gets on this running wheel, and in fact, after the first few days, you can predict when the animal is going to get on and off of the running wheel with precision of 2 or 3 minutes, which is for me, really quite remarkable, imagining that an animal is in constant darkness, deriving this information apparently from something endogenous. And of course, the other evidence for this being an endogenous pacemaker of some sort is that this is not a 24-hour rhythm, it's a circadian rhythm. It's about 24 hours. It's in fact a little bit shorter than 24 hours, for which reason there is a regular advance on each day of the onset and offset of this activity. So both of these features then indicated many years ago that there was an internal mechanism responsible for this wake-sleep behavior in this organism and in the many other organisms that were followed thereafter. So in the 1970s, there were attempts to locate the anatomical source of the cells responsible for generating this kind of behavior. And work in several laboratories identified a small region of the hypothalamus called the suprachiasmatic nucleus, or the SCN for short, which in animals that were given a radioactive form of deoxyglucose showed rhythmic uptake of that material in a night-day fashion. So on the left, for example, you see in a coronal section of the brain of a rodent, this is, I believe, a hamster, you see the SCN densely labeled with this radioactive material, so a high level of metabolic activity, a high level of consumption of glucose. And at night, in contrast, just a trace of activity evident in the same cell group. So this indicated a locus of biological activity, but was this a region of the brain that had something to do with the locomotor records that I mentioned? And a subsequent set of experiments deliberately lesioned that region of the brain, extirpated, removed the SCN, and with the result that those animals became arrhythmic. They no longer showed rhythmic activity just from lesioning this one small region of the hypothalamus. Even more hypothalamus, even more surprising was a later surgical study in which a fetal SCN was transplanted to replace the lost SCN in that arrhythmic animal. Rhythms were restored. So clearly this is a tissue that is fundamental in its contribution to this rhythmic behavior. Now, at about the same time that those experiments were going on, a group at Caltech in Southern California, it was Seymour Binzer's group, who was is well known for his early work in microbial genetics, viral genetics, later became interested in neurogenetics using Drosophila as a model organism. And his student, Ron Konopka, wanted to do something that was similar to the SCN experiment, but at the genetic level, at the gene level. Could he identify not a part of the brain that was responsible, but a gene that was responsible or contributed, was necessary for circadian rhythmicity? And Ron chemically mutagenized a number of strains of Drosophila, and after searching through many hundreds of flies, found 3 different classes of mutations, all of which seemed to affect the same genetic locus, which he named period. One mutant had no rhythms at all, so it was like an SCN lesioned animal. Another had a 28-hour rhythm, a long period rhythm, and the third had a short period rhythm, a 19-hour, a fast-running clock, a slow-running clock, and the loss of clock activity. So it's hard to imagine that there would be any other way of interrupting a clock, but the fact that all 3 classes of mutations seem to affect the same functional gene suggested that this gene would have a very deep involvement in circadian machinery, a gear or a cog in a so-called biological clock. Now, at the time that Ron was just finishing up his PhD at Caltech, this was his dissertation work, and this came out just as I was beginning my own work at the University of Texas in a genetics lab that was interested in another problem, which is, why do animal chromosomes have so much DNA? The typical chromosome in an animal has 25 times more DNA in a fly, 25 times more DNA than one would think it would have given the numbers of proteins that were identified in the animal. And as a part of doing that study, we were looking at these giant chromosomes from the salivary glands of the fly. These are the palatine chromosomes that have been a geneticist's boon for so many years. And we were trying to count every gene that was in this region. And Ron and Seymour's paper came out, and we realized that this new locus, this period gene, seemed to map close to, possibly within the region that we were studying in detail. And so as a graduate student, I asked Ron to send those mutants and was able to use a series of chromosome rearrangements, deficiencies, duplications, and translocations to locate the period gene. And the period gene was found between 2 existing, formerly known genes, both of which were vital to the life of the fly. And this gene sat right between them, and what we realized was that the only phenotypes associated with mutations of that gene, this new gene period, were circadian phenotypes. Again, reinforcing the idea that this gene had a deep role in the specification of circadian rhythms. Another it turned out to be another very important finding was I identified a translocation, a chromosome rearrangement that involved this region of the chromosome and joined two different chromosomes together that turned out to be a period gene mutation. And the reason that was an important discovery is because it gave us a way to get this gene through cloning a few years later in 1983 and 1984. All of Ron Konopka's mutations were point mutations, which in those days you would never identify in a walk through these chromosomes, but a chromosome rearrangement, the break point of a chromosome rearrangement could be easily identified. So in 1984, our lab and our colleagues at Brandeis, Michael Rosbash and Jeffrey Hall, at about the same time arrived at the isolation of this gene, period. And moreover, we were able to, using those rearrangements, identify the gene and then confirm that this gene was, in fact, the period locus by moving it back into animals that had no rhythms, that were mutant for the per locus, showing that we could restore a behavior to an animal that was behavioraless due to a genetic mutation. And of course, that was the equivalent of replacing that SCN anatomically, but now we're replacing a gene and getting hopefully to the bottom of the machinery that was responsible. In the subsequent years, we decided that we were not going to learn nearly enough about a biological clock from studying a single gene, that we needed to screen for many more mutations. We set out to isolate new mutants, not because they would interact with the original mutations of PER, but just looking for other genes that would affect the circadian clock. Whereas Ron Konopka had found his first mutants in the first 200 strains, my colleagues at Rockefeller, Amita Sehgal and Jeff Hall, had to go through 7,000 strains of flies. And this is a photoshopped image of what a 7,000 strain experiment would look like. It's pretty overwhelming. But they did come up with a new mutation, which was arrhythmic. It had no rhythms. We named that mutation timeless for its lack of rhythmicity. But the most important thing about timeless was that when we asked about the phenotype with respect to the action of the period locus, terribly important to us at the time because we had not asked for a gene that would affect period. We were just asking for a gene that would affect the clock. And yet the first new gene we find works with the original gene. And this was our first hint that this kind of an approach could completely describe all the molecular components of this circadian clock. We went on to a much faster way of screening. In this apparatus, you place a single fly inside a small tube. It runs between a phototransistor and a light-emitting diode, and information is collected on a computer so that we could screen tens of thousands of flies per year for mutants. And the results of that was to produce, this is the simplest version of a model in which several components are interacting to produce an oscillator, a molecular oscillator that we now know is the basis for this circadian clock. So the way this system works is that we have two genes sitting at the center of this mechanism. One is period and the other is timeless. Both of these genes are engaged by two transcription factors working together. One is called clock, and which is represented with the C, and the other cycle, represented with the B. And whenever those proteins are in the nucleus, they can drive transcription of PER and TIM, of timeless. Those can be translated to give period and timeless proteins. But now the first of several breaks occurs in this system, and that is that if there is light in the system, if the sun is up, There is another component that was identified by screening, genetic screening, which is called cryptochrome, which is a photoreceptor that when activated by light will bind to timeless, cause it to be phosphorylated and degraded. In the absence of timeless, PER has a partner called double time, again coming from these genetic screens. And double time is a kinase that phosphorylates period when it is alone in the cytoplasm. If there is no timeless in the system, double time will bind to period, phosphorylate it, and period will be destroyed, which means that all day long we have no timeless because of cryptochrome and we have no period because of double time. When the sun goes down, period and timeless now can form a new partnership, which is a per-Tim dimer. And period and timeless now take on a new kind of activity. As monomers, they're stuck in the cytoplasm, but as a dimer, they can move to nuclei. And in nuclei, they physically associate with cycle and clock, which were causing transcription of period and timeless, and cause clock and cycle to fall off the chromosome, so there's no longer any transcription. So we go from a day-night oscillation in the presence or absence of period and timeless, and during a day-night cycle, we can see how there will be cycling of this molecular machine. On the other hand, in constant darkness, this process still works because it takes time to build up timeless as RNA is accumulating, and double time is constantly present, constantly removing period, so there's a delay in the appearance of these per-Tim heterodimers, providing one of the many delays that produces an oscillation in this form of feedback. Now, this is not, of course, sits at the bottom or at the top of oscillations within these systems, but there are hundreds of genes that are cycling in response to this activity. And these are data from 2001 that we performed, in which this panel shows 150 genes stacked on top of each other being followed over a 6-day interval, in which every 4 hours we're measuring the concentration of RNA coming off of that gene. And we've stacked genes that have the closest patterns to one another. We've stacked them closest together. So genes that are turning on at dawn, are peaking at dawn, are shown at the top. Those that are peaking at dusk are shown in the middle, and so forth as we go from top to bottom. And what I hope you see are 6 stripes, alternating green and red stripes, which are indicative of the rhythms that all of these genes are producing. So this clock that we see is really a driver of rhythmic gene activity, in which hundreds and hundreds of genes are being regulated in this fashion. And they're being regulated not only in the cells that are being followed here, which are from the head, but all over the body. One of the things we realized that these clocks are operating in our skin, in our muscles, in our liver, in our brains. Most of our cells carry independently operating clocks driving these kinds of gene expression rhythms. Now, another thing that we learned in doing this is that if we look at our mutants, timeless clock or period mutants, we find that this pattern of rhythmicity disintegrates. All of the rhythms go away. So without this central oscillator, without this central clock, none of this gene expression rhythmicity persists, even in the presence of a light-dark cycle. So if we think of this in evolutionary terms, we know that these clocks evolved in response to cycling light and temperature, and yet once this clock has developed, it has taken over the system to the point that without the clock, even the original environmental changes that drove the evolution of this system can no longer provide changes in gene expression that are so essential for function. So let me now, with that background, tell you about our work on human biology. We began this about 5 years ago, and we were interested in a syndrome called delayed sleep phase disorder, or DSPD. And this is a cartoon that was produced for us by our communications department at Rockefeller, and I think illustrates well the problem which many of us have, which is in the middle of the night, we're up staring at a computer screen, can't get to sleep. This is not a facultative problem for carriers for those that have this disorder. These are chronic night owls. So these are individuals that have a very difficult time getting to sleep at night and a very difficult time getting up in the morning. And I'll show you examples in just a moment. But here are some of the key features. First, it's fairly common. About 5% of the population in the United States appears to be affected by this kind of a disorder. There's a persistent delay in the timing of the major sleep episode and strong resistance to efforts to control that pattern of sleep. Alarm clocks don't do much good for these individuals. Now this slide shows an example of a control individual at the top, the sleep-wake pattern, and a DSPD individual, which will be a proband for a study I'll be telling you about on the bottom. And what we're doing is we're double plotting the behavior. We've given them an activity watch that's like a wristwatch that they wear continuously, even in the shower. And they also are given a log to write down when they get out of bed and when they go to bed. And the in-bed times are indicated by the red triangles, and the getting out of bed log is indicated by the blue triangle. So you see our control has very steady behavior, a very steady bedtime, a very steady wake time that we would all aspire to. On the bottom, though, we have a DSPD individual, and it's a very ragged record. It's difficult to pick out a pattern, but if we do ask about in-bed times, we find that almost always the in-bed time is well after midnight, and most often around 4 in the morning, and the wake times are typically just shortly before noon. So this is an individual with a severe sleep disorder that they've come into the clinic to try to understand better, see if there's any help. So it turned out that the individual on the bottom had a family that included other members that had the same complaint about their wake-sleep behavior. And what we did with this small family is whole exome sequencing of all the members of the family that were participating, and we found a single gene that was shared by all of those who exhibited a sleep-wake disorder, and that was a gene called cryptochrome 1, and I've already introduced cry on a prior slide. The mutation is a simple nucleotide change. It's just downstream of one of the exons. It's in what we call a splice donor site. The reason that it has such a great effect is that this mutation removes that splice donor site and causes a downstream exon, exon number 12, to be spliced to exon number 10. So exon 11 is missing in carriers of that mutation. And we see that happening down here if we look at the RNA being produced by this individual, which is individual number 11, as compared to several other members of this subjects that were in this study. Subject 11 is making 2 forms of the RNA, one from a wild type, a normal copy of the gene, and another truncated form that is reduced by the expected 72 bases in length. And below this is shown the protein produced by the same group of individuals, and once again you'll notice that individual number 11 has 2 versions of the protein that differ by 24 amino acids, the wild type copy and the mutant copy. So all of the individuals that showed the syndrome were heterozygotes, which allowed us to conclude that this is a dominant mutation. It just takes one copy to have this severe night owl sleep disorder. We found that we could take that version of the gene and we could transfer it to cultured cells that had no cryptochrome. And we could restore rhythmicity in both cases with either the wild-type version or this variant mutant version that we found in the family, and that the, as shown here on the lower left, the mutant form of the protein restored rhythms with a longer period rhythm than the wild-type form. And this, again, is in cultured cells. We also found that the skin cells of these individuals were showing long period rhythms. So this was confirmed that this is a hardwired disorder that can be attributed to this particular gene. So let me tell you a little bit about the human clock, because there's a subtle difference between that and the fly clock that we originally worked with. It's got all the same components, but there are some slight differences in the way the system works. Instead of having timeless as a partner for period, cryptochrome in us, in humans, is the partner for period. So cryptochrome is a transcription factor that pairs up with period, moves into the nucleus, and drives clock and cycle, which are known as clock and BMAL, off of the promoters of the period and cryptochrome genes to close this molecular cycle that we were talking about before. So we have a different set of questions now about this mutation in cryptochrome. How does it affect the ability of this inhibitory system to do its work on the period and timeless loci, on the transcription factors that ordinarily cause transcription off of period and cryptochrome? So the first question we asked was whether or not there were differences in the physical activities, the physical interactions between these proteins, since that's what we knew was driving most of their function within the animal systems, all of the animal systems that have been looked at. And I'll just start by pointing out that what we were able to do is to transfect cultured cells with all of the components for the transcriptional machinery, clock, BMAL, cryptochrome 1, and period, and tag these in such a way that would allow us to look at the relative affinities of these proteins for one another. And so down here in the lower part, what we're doing is we're immunoprecipitating a tagged version of clock in the presence of either the full-length form of cryptochrome, the truncated form of cryptochrome, or a 50-50 mixture of the full-length and truncated form of cryptochrome. Now over here is shown the input in this experiment, and over here are shown the results. And what you see is the full-length form of the protein is very weakly recovered in these assays, whereas the truncated form is robustly recovered, and when we have a 50-50 mixture, it's almost all the truncated form of the protein that is being pulled down in association with that truncated form. So I can go further into this. I'll just tell you what the rest of the data say. They tell us that period and cryptochrome, there's no change in their affinity for one another, but there's a huge affinity change for the period cryptochrome complex and the clock BMAL, or clock cycle complex, with the truncated form essentially pushing the wild-type form of the protein off the promoter and replacing it. So what we conclude is that this is a hyperactive transcriptional inhibitor. It sits on these genes, turns them off, and keeps them off for a longer duration on each cycle, lengthening the cycle to give a longer period. Now, for us, the most fascinating piece of this study was when we started to look at the genome databases to find out what the frequency, the worldwide frequency of this mutation might be. And for this audience, I think you're probably interested in the European frequency of this mutation. And in fact, the frequency is so high that it suggests that about 1 in 75 non-Finnish Europeans is a carrier of this gene, which means, and you only have to be a carrier, you only have to have one copy of this gene to show this sleep disorder. So this is quite a prevalent sleep disorder, something that we certainly didn't imagine when we began the work. There are some populations where we can't find the mutation, and there are others where it's quite abundant. And I think this is telling us something about the origins and the spread of this mutation in natural human populations. Because this was so prevalent, we set up a collaboration with a group at Bilkent University in Turkey. Tayfun Özalek is our collaborator. And we began to look at other families. We began to search for families in DNA databases that were carriers and then phenotype them with regard to their sleep. And what we found were 9 or 10 families. A few pedigrees are shown here, but most importantly, in the lower right, are shown the results of the sleep phase of these individuals. So MSF, or mid-sleep phase, is just taking the center of the time during which each of the individuals are sleeping each night from their sleep logs. And what we see is for all the non-carriers, they have nicely performing mid-sleep points at around 4:30 in the morning. But most of the carriers, heterozygous or homozygous carriers, have mid-sleep periods that are mid-sleep points that are delayed by about 2.5 hours, suggesting that they are going to have a delayed sleep pattern. There's another pattern, though, that we see only in the carriers, and that is a fragmented sleep pattern. These are nappers, and they take multiple naps around the clock. It's a very odd pattern of sleep and rest that we're quite interested in exploring further. So I can summarize what I've told you so far, that we've got several unrelated families where we've studied this genetic variation, and its presence clearly predicts the DSPD, delayed sleep phase disorder, and the penetrance and frequency of the allele certainly suggests a broad contribution to this disorder worldwide. Since the mutation's found in multiple families, it's being transferred, it's being inherited within these families, and therefore every tissue in their bodies must be carrying information for an altered clock. So we're very interested in what besides sleep is affected in these subjects. As I said, there's enhanced binding of this version of the protein to its targets, so it's a strengthened transcriptional inhibitor. And the competitive binding suggests a basis for its inheritance as a dominant trait. And finally, we've shown by gene transplantation that it will lengthen, that this gene alone is sufficient to lengthen the period of a naive cultured cell. So let me tell you a little bit about where we're going with this. One of the things that we've done is we keep up with the database, especially the one that's maintained at the Broad. This is an exome and whole genome, whole exome and whole genome combination database that is now, as you see at the bottom, composed of about 140,000 genomes or whole exomes. Now, and as you see, we again have these large differences in distributions of this particular variant in those databases. But the point I want to make is that as these databases grow, already a sequence database that's composed of 140,000 whole exomes is already going to have most of the heritable human variation that's going to be associated with clinically significant disorders of sleep. If it's not found in 140,000 exomes, whole exomes or whole genomes, it's going to be so rare as to have to require an inordinate amount of effort to study or think about a therapy. For most of the genes in these databases, there are identified polymorphisms every 3 to 4 nucleotides. You can sit at a computer screen and pick a gene that you know something about, and you can see all the variation that's been seen to date in that gene. It's an extraordinary list. And the allele counts and the population-specific carrier frequencies are all given in these databases. So what we're doing is we're focusing on candidate polymorphisms that are both prevalent and predicted to have an impact on the clock. Again, because we know so much from model organisms about the way the clock works and individual proteins function, we're able to pick mutations that we think are very likely to have a significant clinical impact. And of course, then the molecular mechanisms for candidates can be explored in cultured cell models. And again, the prevalence allows us to find subjects to test those models by direct phenotyping of human subjects. So this is a list. RS numbers referred to, each of these RS numbers refers to a single mutation found in the databases for several of the genes that we study: clock, PER1, PER2, PER3, CRY1, timeless, CRY2. These are all mutations that have what appear to be impactful mutations, and where we also have subjects involved in more than, in multiple families in the Middle Eastern database that we're using. So this is so much easier than studying genetics in Drosophila, in fruit flies. It's an extraordinary time to be studying human biology. There are other questions that we have to ask about these. So even the gene that's in hand, the CRY1 mutation, there are so many hypotheses where there are anecdotal links between circadian rhythms or sleep and disturbances of clinically important disorders, mood disorders, metabolic disorders. With these higher allele frequencies and the identification of large numbers of subjects to study, we believe we can make great strides in assuring ourselves that anything we see as a comorbidity is actually genetically linked, actually has a genetic basis. And I think this is for the first time a way of sorting through these anecdotal associations and putting them on firm ground. And again, these would be expected to be reflections of the fact that these mutations would have effects in multiple tissues. Finally, I can just give you an example of the kinds of information we have by going back and forth between a model organism and our human biology. We know from work by others that CRY1 plays a central role in the regulation of gluconeogenesis in the liver. This is from mouse work. There's a mouse model in which constitutive overexpression of CRY1 blocks glucagon signaling in the liver to produce hypoglycemic mice. And we also know that in wild-type mice, there are oscillations in CRY1 that periodically inhibit gluconeogenic gene expression in synchrony with day-night feeding fasting cycles. So we've now described a dominant gain-of-function mutation of CRY1 within a large number of families. This is a dominantly active transcription factor. It's an overactive gene. So we have a simple question: does CRY1, this mutation, alter glucagon signaling in human carriers? So these are the kinds of informed questions that we can ask, again, by moving back and forth between model organisms and human biology. So again, I think the prospects for learning rapidly about human biology, not only in this sphere, but just due to the strength of the databases out there, and the more we know about particular molecular pathways, the better chance we are to make educated guesses about which variants are going to be the most interesting to study. Finally, let me just close by thanking the people that actually were doing the bulk of this work. Alina Patka is a postdoc in the lab that has done all the molecular biology, helped assemble the team that produced this work over several years. It began with a collaboration with Weill Cornell Medical School next door with Scott Campbell and Patricia Murphy, who identified the subjects that we were able to work with. Anna Krieger, also at Weill Cornell, helped us analyze the sleep records from those individuals. And Tayfun Osalek and his student Onur Onat were responsible for helping us carry this to many, many families in their Middle Eastern group. And finally, I think credit goes to our friend Drosophila, where all of this started. And I can't tell you how pleasant it is to start with a fly and after 40 years be working on the same biology in humans that I feel was critically dependent on the work that came from the fly to move forward. So I want to thank you all again for this wonderful presentation, for this honor that I've been given. And I hope you remain as excited as I am about the future of human biology and clocks. Thank you.